All right. Good morning and good afternoon, good evening, depending on where you are in the world. This is the second workshop in a series that we started at the last Biocuration meeting on the intersection of machine learning and biocuration that has been organized by myself, Lynn Schrimmel, and Cynthia Smith. I am just going to put an initial plug in here for the ISB, which is the International Society for Biocuration. Our annual meeting is October 18th. And we're going to have two talks by our Biocuration Career Award winners, Nico and Charlie, who will be talking about their experiences in biocuration. And if anybody is interested in coming to that, you can say something in the ISB Slack that we sent you a link for in the registration, and that we can also drop an additional join link into the chat.
Done.
Yes. So the goals of this
that's
workshop.
On Zoom. That's on Zoom, by the way. Sorry.
That's on Zoom. Yes, the Zoom chat has that link. The goals of this workshop are to try to initiate dialogue between the biocuration community and the machine learning community to try to really further these collaborations and improve and enhance the ways that we can work together to bring machine learning and AI and large language models into biocuration to really improve our efficiency and hopefully then also improve the tools that are being produced out there and hopefully make research go that much more efficiently. Our speakers today are Fabian Neuhaus, Robert Lehman, and Robert Hohendorf. And the first speaker will be Fabian, and I will stop my share and hand it over to you. And at the 14-minute mark, I will turn my video back on to give you a warning that you're almost out of time.
And we encourage people to post questions in the chat or the Q&A. Sue and I will monitor it, and we'll facilitate those at the end of the talk.
Yes, thank you, Lynn. I'll also monitor the ISB Slack channel if you want to put questions in there that maybe we can discuss later.
All right. That's bizarre. Okay. Do you see my screen?
Yes, looks good.
Okay. Thank you very much. Yeah, thank you very much for the invitation. My name is Fabian Neuhaus. I'm from Magdeburg, Germany. And I felt like the introduction implied that I'm from the machine learning community. I want to not pretend I am. I would feel like a fraud. My background is in ontology. I'm a philosopher by training, really. But I will talk about machine learning today and about the synergies about how machine learning can help us to develop ontologies and use ontologies, and also how ontologies can be used to help machine learning tasks. And let's start with the first question. And the context is, on all my examples will be from CABI. I hope that many of you are familiar with CABI. CABI is an ontology for chemical entities of biological interests, and it contains information like the chemical structures, some ID, names, annotations, like most ontologies. You will be familiar with them. And CABI faces a challenge that CABI is not small. CABI, the fully annotated part of CABI, is about 60,000 entities, but PubChem, for example, contains 100 million entities. So if you think about the fact that CABI grows about 100 entities per month, it's clear that CABI will never even cover a significant chunk of its intended domain. Now, that leaves us to a dilemma. On the one hand, the success of CABI and many other biological ontologies the success depends on their quality, and the quality is achieved by manual curation. And I won't go into details, but I'm convinced that that can't be automated. Can be discussed maybe in a different talk, but let's assume that it can't be automated. But the example just illustrated that ontology development doesn't scale for certain areas, in particular chemistry. So what do we do? And one approach is to automatically extend the scope of a manually created ontology. So the architecture is like this. You have here some chemical formula, some structure, and you want some magic tool that basically takes that and says, okay, this is not in Cabby, but if the ontologists who are developing Cabby would extend Cabby, they would put it under purine alkaloid, right? So you basically predict the classes, the superclasses of that particular class of that is represented by this chemical structure. And there actually such a tool is already exists in a rule-based form, which is actually used by the developers of Cabby. And we developed here in Magdeburg various versions of such tools with machine learning tasks. And so there are different encodings you can use, and there are different kind of classifiers you can use. We trained that. The important thing that I just want to point out is that this is only trained on the content of the ontology. So we don't use external sources like text corpora. So basically, we try to train a system to recognize the decisions that the ontologist made, which is implicitly represented by the structure of the ontology. And to make a very long story short, LSTMs, which is a particular kind of neural networks, performs in general better than traditional approaches of machine learning. And you can, then we could also use transformers, which are the kind of neural networks that, for example, these large language models use, like ChatGPT, and they even perform better. And as a result, we get something like an F1 score of 0.9 something for the best performing models that we have so far. And if you want to try it, this is available as a tool, but also we have a small web interface where you can just upload some arbitrary chemicals, and then it tells you where it would fit in Cabby. Now, I oversimplified a lot, and didn't mention some of the limitations, but just one of the limitations. So not all classes can be learned equally well. Some classes are notoriously difficult for all kinds of approaches. So, for example, chemical structures have often these ring structures. If you think of what the neural network sees, it sees something like this flattened string. And it's very hard for a neural network to learn that it needs to basically connect this string and traverse it in order to do the right classification. That's an example for difficult stuff. But and no method works best for all classes. So something we want to work on in the future is an ensemble approach where we combine different methods. But just to take a step back, what does it show? So the idea here is that this illustrates that, well, one way of using machine learning is that we can use a manually created ontology of a high quality and then extend basically its leaf nodes automatically to stuff which isn't covered, either automatically, or you can also, of course, think of it in a semi-automatic use case where you, as a developer, just validate whether this prediction actually makes sense. One other thing which is very, I think, positive about this approach is that you get a feedback loop, a positive feedback loop, because if CABI is extended, then we can retrain the model, and the performance of the model will be better. And we also can just by looking at the training data, see which classes are not particularly well trained yet, or where, you know, performance is bad. So you can, if selectively decide to provide extra examples for those, and then extend CABI manually a bit in this direction, and help the machine learning tool then along to have better performance for these classes which don't work so far. All right, so this is the one direction where we can use machine learning to help ontology development and annotating of data. Here's the other direction is, can ontologies support machine learning? And for that, I want you to be, for the moment, be my classifier and classify a Woblox. So this is a typical training situation. You get some training data. The first monster is a Woblox. Second is no Woblox. The third is a Woblox. And your task at the neural network is now to predict, is the fourth guy, the blue guy, is it a Woblox or not? And this is obviously a very hard task, because there are so many things you could pay attention to. You know, size, shape, color. You can count the eyes, right? Or the teeth, right? Maybe presence of teeth is important. So this is the problem for machine learning. Too many things to pay attention to. Now, imagine I give you an additional information. I tell you that there is a monster ontology where the world-leading experts on monsters created a classification scheme. And the one major classification is between monsters with horns and monsters without horns, because it turns out that that is a tremendously important distinction to predict all kinds of other things. In this case, knowing that, having that background information, looking now again at our example, you might be inclined to see, okay, my positive examples for Wobloxes have no horns. My negative example for Woblox has horns. So maybe that would help me to predict, no, that the blue guy is not a Woblox. Of course, you can't be sure, but it helps you to make maybe this, you know, focus on this particular aspect of our monsters. So that idea that you basically look at an ontology and say, even though the word Woblox doesn't appear here, the ontology is basically a record of what classifications have been useful in the past by experts in the domain. And so if we can, if we now start to talk about new features in the domain, maybe the stuff we have talked before, these classifications we had made before, maybe they are predictive for other future tasks. So that's the basic idea that we exploited. And here's how we exploited it. We basically used the same model I just talked about, the one which allows us to predict classes in CABI. Because what does it mean? It means the model kind of learned aspects of the ontology. It learned to group things together by their chemical structures and, for example, recognize alkalides. And then the assumption is that these chemical structures may be predictive for other things. And we tested that with a toxicity prediction. This is just because there was a data set that is available. Tox21 has 12 kinds of toxicity. The setup is then that you, again, get a chemical structure. The system that has been pre-trained previously on ontology class prediction is now retrained to predict toxicity. And because it has background knowledge from the ontology, it actually can use that to talk or predict toxicity. And it does that actually quite well, significantly better than without this ontology pre-training. These are I don't want to go into the details, but basically the graph, the blue graph here, or the blue line, represents the with pre-training, the red line without. And as you can see, the training is quicker and more efficient. And it actually turned out that our model with this ontology pre-training outperformed the state-of-the-art of other more chem informatics-based approaches, which are specific for toxicity prediction. So this example shows that by using the ontology and making the system predict classes in the ontology, we can actually train models to understand, in quotation mark, some of the semantics that is represented in the ontology, and then reuse that background information when for training tasks which have nothing to do with the ontology. I mean, a side note, Cabby has some information about toxicity in there, but we didn't use that part for this particular experiment. So it really, the part we used was just about structural classification. Now, I want to address transferability to other domains. All the examples I gave you were now Cabby, and the relation, or we use the SMILES representation, which are part of Cabby. So the question is this approach limited to chemistry? And the answer is no. For us as people in the computer science department, we don't have domain data. Cabby is particularly convenient because we could use Cabby to generate the data sets that we need for the machine learning task. You know, as computer scientists, we always have the challenge of getting good data from domain experts. So that was easier to just work on that. But in general, all you need is an ontology, preferably a good one, and then some data which allows you to automatically classify, or you train a model to classify the entities in your domain with labels from the ontology. And if you're doing that successful, then the resulting model will have absorbed the semantics or some of the semantics of that ontology, and hopefully will be able to perform other prediction tasks more productively.
Can I ask a question?
Well,
just
just let me finish.
Type them into the Q&A, please, and then we'll hold them off to the end.
I'll be 2 minutes. So in summary, machine learning can support ontologies or ontologists and the users of ontologies by because you can build classifiers which can learn to extend ontologies, and we can build tools like the Cabbyfier, which or we have built such a tool which enables Cabby developers and Cabby users to classify chemicals. And ontologies can be used to guide machine learning. By ontology pre-training, you can make the model to observe some of the semantic connections you have in the ontology and then reuse that for other training. This work is a joint effort by many people, in particular Martin Glauer, who works with us here as a PhD student and writes his thesis about this, and Jana Hastings, my good friend and colleague from Zurich, who is, of course, the expert on Cabby. Thank you very much.
Okay, thank you, Fabian. Yeah, so we have now about 5 minutes for questions. Christian, if you want to ask your question now, you can unmute.
Okay, yeah, 2 questions. First, so your training, you can predict only leaf nodes for Cabby. Is that what you said? You can't predict, where a term or a molecule go in in between nodes, in between leaf and parent node.
Let me see whether no. Well, no, quite so what we do in this particular, because we basically use Cabby and use the higher part of Cabby, the top to the medium level as the tags we want to predict, and then we use the lower part of Cabby, all of the specific molecules, right, which have SMILES attached with them, to for training. So what we predict are actually the mid-level and higher level classes. So for example, here, this wonderful chemical is classified as benzene. Does that answer the question?
Yeah. And then the other question is, can you have multiple relationships between the nodes, right? Not only is it, but also other relationship between nodes. Would you also take them into account? Or is it depending on what kind of question, what kind of thing you want to predict?
So what it predicts is classification. So it's only currently predicting subclass relationships. And for the training so far, we have used mostly the version that is online live right now has been trained on only the subclass relationships on the part of the ontology. We are currently having a master student who's now using the roles, like chemical roles and biological roles, which are also part of CABI. And but that isn't available yet. That's still running. And yeah, I could go into more details, but yeah, it's a bit complicated to use more than just the subsumption and the disjointness axioms.
Okay, thanks.
Yeah, Lynn, do you want to ask your question?
Yeah, I had a question. So if you're going to integrate these predictions into the ontology, have you thought about how you'd want to note that? Do you foresee in the future an ontology that has manually curated nodes and subclass decisions and predicted ones together? And then how would people distinguish them?
So CABI itself, when I said it consists of 60,000 fully annotated nodes, then I should say for completeness, it actually consists of 140 or 160,000 nodes. The other 100,000 nodes or entity classes are automatically generated already by a tool which is called Classifier, which is functionally equivalent. It just uses a rule-based approach, and there are other differences. But that's basically our comparison. So if you look at CABI actually consists of partially manually curated parts, and then it's extended by an automatically generated part. Now, I don't think that I don't think I would want to do this in that way. If it was my decision, I would just say you have your ontology, and then you offer a service where you just, if you want to have for the automatic generation, that's our approach for CAVIfier. So it's just an online service, or you can also download it and run it on your machine. And if you so you can basically create classifications on the fly. But of course, if you then, I guess, if you want to reuse that, then you would have to put it in some extension of CABI. Yeah, but I personally would clearly distinguish between manually curated and extended nodes.
Okay, thank you very much. We're basically out of time. So thank you. That was a great talk. And we will move on now to Robert Lehman.
All right. Let me share my screen here. So we all see this? Okay. Everybody hear me okay? Audio doing okay?
Looks good. Yep, it's good.
Yes, my name is Robert Lehman. Thank you to the organizers for inviting me. I'm a staff scientist at NCBI, working with Dr. Jiang Liu on natural language processing. Basically, no surprise to this group, biology's an information heavy field, even becoming even more information heavy, medicine too for that matter. We get this sort of information ecosystem And, you know, while the literature is important and near and dear to my heart, being at NCBI, as the literature services are, this information reuse from the literature itself manually isn't, you know, it's not computable, it's not automatable, it's not efficient compared to what you can get with the databases. And so one of our goals with the biomedical natural language processing is to support biocurators and with tools that partially automate these semantic tasks. And one of the most fundamental tasks is named entity recognition, represented here, where with chemicals and diseases that are located within the text. And then the next layer of complexity above that would be the entity linking or normalization, sometimes called grounding, you know, where those entities that are located then are identified as specific concepts within some sort of vocabulary or an ontology. So those two combined, along with a third task that's relation extraction, identifying relationships between these entities that appear in the text, sort of form a sort of a foundation of basically together they're biomedical information extraction. And you can power a lot of if you have with good quality support there, you can power a lot of really interesting tasks with that, everything from, you know, semantic search, biocuration, of course, all the way through like literature based discovery. So at this point, given all of the interest in the last year or so, and you know, even family members who have no clue what I do for a living asking me about ChatGPT, need to mention, hey, can we just, okay, I want to get this information out of the text. Can't we just ask ChatGPT to do it for us and don't have to mess with all of this? And the answer, as you know, I've got a rest of the talk, but I have to tell you, at least at the start, the answer is yes. If you don't mind the performance that, you know, sometimes, you know, the automated tools aren't quite good enough, but the performance of ChatGPT, you know, you're getting twice as many or even more than that errors from ChatGPT than you would with a just a, in this case, it's an off-the-shelf PubMed BERT system. These are 8 different tasks representing a broad range of what you know, tools can, what current natural language processing tools can handle quite well. And the other challenge here with this is that is the cost. So just the ChemProt test set required over $1,000 to run with GPT-4. So in this case, you know, it does become a real problem. Having said that, there was one area where GPT-4 really did shine, and I apologize if there's any noise that causing some issues, people outside. But in that where GPT really shined was with the question answering task. It is trained as a chatbot after all. So, okay, so if that's the case, and we can't just throw ChatGPT at it, at least not without thinking maybe a little differently about how we want to do it, what are the problems? Well, at a high level, there's 2 problems. The first one is variation, where you have many different ways of saying something. This is just an example of the different patterns that are used for naming diseases. And the problem isn't even so much that there's multiple patterns, it's that you can have the same disease have synonyms with the all of the different patterns, so that you end up with, you know, a single concept or arguably a single concept blowing up to you know, dozens of different synonyms, and we have to be able to identify all of these. And of course, the vocabulary changes over time, but we, with text mining, still have to be able to extract information reasonably accurately from things that are, you know, from ways of saying things that are really not in use anymore. So that name variations is a real issue. We found this particularly to be the case with a project recently that we did on long COVID. We have a system for putting out the lit COVID, for putting out the research on COVID, and we found that, you know, there's these long-term effects, but we originally weren't capturing those articles, and so we needed to do that. But in order to do that, you have to be able to identify where people are talking about, some sort of long-term effect due to COVID, and there, we identified, these are just the top ones. As of last night, I believe I checked, it was 3,800 different ways of saying long COVID. And of course, the huge diversity here is that there really wasn't enough time for a consensus to develop before the interest developed. And so you get all these articles that are talking about it, but they all have different ways of saying it. And so you can see also on this log chart, most of the variation, most of the phrases that mean like a long-term effect really only occur just a few times. And then of course you have ones that appear very, very frequently. But that's just variation. We also have problems with ambiguity. Of course, the stars of the show for ambiguity are the TLAs, the 3-letter acronyms, here seen with the example of SCP. But authors tend, not always, but they tend to at least define those once in their paper. So there's ways of handling this. But believe it or not, that's not the only problems that we have with ambiguity in the biomedical literature. If an author says that they're working with yeast, are they working with the model organism? Or are they working with one of the 1,500 other species of yeasts that are defined, including Candida auris, an emerging pathogen? So ambiguity is also an issue that we have to deal with. So how do we deal with these? We've already seen transformer models briefly. That's basically one of the most transformative things since I got my PhD. Has been these models that we start with a large corpus, and we essentially, this model is trained to, given the context, predict a word. And it's trained with a very large amount of text, preferably text from that it will be seeing later. So if you're looking at biomedical publications, it needs to include biomedical publications, or if it's EHR text, it should include that. And then once you have that, you have a trained language model that can be used for different tasks. And then, so what we do with that is we take a manually annotated data set that represents that task, and we fine tune that language model to perform that task. And from there, of course, we can apply unannotated, we can predict that task on unannotated documents to give some automatically annotated predictions there. So, fortunately, the fine tuning is not too complicated. This pre-training step is very expensive, but the nice thing about it is that once you have that, if it's done well, you can actually fine tune it for different tasks. So, we have systems that use language models, typically transformers like BERT, to perform all of the steps, named entity recognition, entity linking and normalization, and also the relation extraction. Our pubtator system is about 10 years old now, and has performed the entity recognition and linking for all of that time over PubMed articles. Later, we added the full text, and most recently, now with the pubtator 3 beta, which is available now, you can see at the link there. It also has relations between the different entities that are mentioned in the abstracts. So we also have an API, which is really important, but I'd like to briefly talk about this tool because it's our hope that the features will be found to be quite useful for biocuration. While the interface itself is good for playing around, probably if you're going to integrate it in a workflow, you won't use this. You'll be using the API, but that's okay. We can talk about the features here. So you can see in the upper right the different concepts that are identified: genes, which includes proteins, and then we have diseases, chemicals, variants, genetic variants, and then species and cell lines. We also, here on the left, have the different relationships between the entities that are found. You can see that there's a relationship here between doxorubicin and KDR. KDR is what NCBI gene calls the VEGFR-2 protein. So we do identify these regardless of, well, attempt to identify them regardless of what synonym that the author uses. And also, up at the top, you can see this article. I found this article using a semantic search. So we search for the chemical doxorubicin, regardless of how the author mentioned it, and also neoplasms, which is what MESH calls cancer or tumors. So that this semantic searching feature can actually be very, very powerful. A couple of different use cases that PubTator has been used for. Semantic search, we mentioned that LitVar is an example of that. Uses the PubTator entities. Other groups have also used PubTator. There's a PubTator, a knowledge graph for plague, Yersinia pestis, and for biocuration directly with Uniprot and Swissprot. And for literature-based discovery, this is just a few examples. And actually, I used it, of course, myself for the long COVID project, identifying mentions of long COVID, but also the mentions of things that are around it, so that I could predict that long COVID was probably related. So, having said this, right, it's important to know annotated data are scarce. So even though the transformer model has been trained over, say, all of PubMed and PMC and perhaps some other things, the fine tuning has to happen on manually annotated data. And this example here is from named entity recognition. If we intentionally, excuse me, overestimate the number of human labeled articles, for biomedical named entity recognition by pooling absolutely all of it, even if it's only had one thing annotated, it'd be only something like 0.1% of all of PubMed would be considered annotated. That's a whole lot of variation that, even just named entity recognition, which is a fairly low level task, has not seen. So this does tend to cause generalization issues. So you have subdomains where things are just a little different. And so the performance tends to be lower in those areas than, say, on the test sets that we evaluate them on. And this is one of the struggles that we have. And of course, we really want to fix it. Got a few ideas for that. The reality is that you're typically, if you're going to use systems in biocuration, there's going to be some degree of adaptation required. These are automated systems. Talked about generalization a second ago. Errors happen. Turns out a lot of errors can be fairly straightforward to fix, right? At the beginning of the pandemic, we had a case where we weren't identifying remdesivir. And so you had to do dictionary post-processing and so on. You can add a little bit of training data. And then sometimes you have to do adaptation, which could either be simple or not. What do we need? Well, domain expertise. I'm a computer scientist. But to truly understand these issues, need to work with biocurators and with bioontologists. You need to know that the number one way for an NLP system to interact with your ontology, no matter how nice everything else is, the number one thing is those synonyms. So they've got to be consistent. Of course, we need annotated corpora, but even just error reports would be very helpful for us. And of course, there's questions about how to, at a systemic level, how do we incentivize, turning these into products and adapting them for slightly different use cases and so on. And I've got a couple comments about trends, but I think we're a little low on time, but I'll mention them quickly. So one of the things that I am seeing is a lot of things can be handled without a whole lot of training data now. So we can do some unsupervised tasks. That's partially because we have more capable models. These models are starting to become like interchangeable parts. where, oh, something new comes out. Okay, pop out PubMed BERT and pop in something else and try it out and see if it works. And that's pretty straightforward. Paradoxically, though, that means that the training data we do need becomes even more important, because it's less about the model, it's more about that data that defines the task. And then, of course, there's questions about large language models, right? The most important thing, in my opinion, to mention about that is large language models, as they currently are far more fluent than they are trustworthy. So they're going to sound like they know what they're talking about, but there's no logic or reasoning behind them, or if there is, it's paper thin. So one of our thoughts is you use them for retrieval and summarization, because that handles their fluency, that makes them shine with their fluency really well. So of course, there's a lot of people involved in this. I especially want to mention my PI, Jung Liu, but also PubTater from the beginning has been my colleague Chishuan Wei's project, and but of course there's many others who have been involved over the years. So thank you.
Okay, thank you very much, Robert. That was a really great talk, and we have actually several questions.
Okay.
I will give you first the one that came from our audience, and then I think I'll let Robert and Lynn ask their own.
Okay.
From our audience, the question is, how is PubTater's performance to identify assays from the literature? And I'm guessing it's specifically expression assays. So how well can you pick out what are the different types of assays that are in the literature?
Okay, so I'm going to start off by answering that we've got 6 different things we look for: genes and proteins, chemicals, diseases, which also includes other conditions and symptoms and so on, even abnormal phenotypes, genetic variants, species, and cell lines. Now, so that is what we identify. For those assays, do those fit into any one of those 6 categories?
Yeah, and I don't know.
Yeah.
I see. I'm afraid I don't know how to.
Right. I don't think so. I don't think so, because we are looking for specific assays, like in the methods. Yeah. Thank you. But maybe you can keep in mind, because that's his need.
There is a need for that. There's a need for treatments. There's a need for anatomy mentions. So that is an interesting one. Then thank you.
Yeah, anatomy actually has been used a lot. Anatomy is, I would say, next priority. But assays, yeah, there's a lot of people looking for that. Thank you. Thank you.
Thank you.
Thank you. All right, and then there are a couple questions from Lynn. One was, how would semantic relationships defined within would semantic relationships defined within ontologies be useful for PubTater?
Yes. Yes. Well, certainly. They would be helpful. There's a couple different places where those would be useful as low-hanging fruit, but they wouldn't be directly used immediately. For example, low-hanging fruit, one way that you can sometimes identify entities if you're not sure what it is by what it is related to. So you'll find things that are around it, like genes. You need to, if you find a mention of the chromosome location, that's a pretty, useful way of saying, okay, well, it could be one of these 3 things, but only one of those is at that location. A simple example. So that's certainly something that would be helpful. And then, of course, going forward, we just now added the relation extraction, and so, but certainly there would be ways moving forward of improving that using resources that describe relations in a large scale sense, in the same way that we are currently using like the UMLS has very, very large vocabularies, and we can take advantage of that to identify, say, synonyms and so on.
Okay, I think we are basically out of time. There are several more questions in here that we didn't actually get to. That is my timer going off. So if you wouldn't mind looking at the Q&A and maybe addressing some of those as we go on to our next talk, which is Dr. Robert Hohendorf. Thank you for joining us.
So thank you very much for the invitation. Can you see my slides?
Yes.
Yes.
Perfect. Okay, so thank you very much for the invitation. It's my great pleasure to talk a little bit about half going to be about research that we have done, and the other half is going to be more an opinion about how machine learning and biocuration will change now. So I have this rather nondescript title. So let me dive in. So we all know that biology, in contrast to many other natural sciences, is a very knowledge-driven discipline. We have these micro theories about pathways, individual pathways or cell types, their behaviors, everywhere in biology or systems biology models. So we have these small descriptions, small words, which are very detailed about how certain phenomena occur in biology. And a lot of this are judgments which come from experiments and they're supported by evidence. So even very simple facts like the function of a protein or whether two proteins interact in a particular way. So we have this, and this is very different from, for example, physics, where we try to reduce most of our knowledge down to some very basic laws, and then we can derive based on few principles and the rest of this field from these very basic laws. In biology, this is not the case, and we need, in order to do biology, we need access to these micro theories, to these small theories or small descriptions of phenomena, and they are derived from hundreds of years of experiments, and most of the data is no longer there. But even if the data is there, we want somehow access to these kind of judgments, to these kind of facts. We want this in order to know this fact for retrieval, but we also want this, for example, for machine learning if we want to have a supervised. And the place where we can find this information is still largely literature, and not even literature or textbooks, but sometimes it's data tables from in literature or in textbooks. So it's not even there; it's even data in a book or in a paper. So the task of biocuration was, in the past, there's a very nice paper that the task of biocuration is to distill knowledge out of text, and it's a little bit controversial that I call this here the past. So I know this, and I will maybe revise this a little bit. But the reason we needed biocurators is that we needed somebody who knows about the domain in order to even understand these phenomena and make sense of the data. So if there's a big table, we need somebody to make sense of it, and this relies on a lot of years of training and a lot of knowledge about this domain. We also need biocuration to be aware of the knowledge bases, at least the schema of the basis of the knowledge bases or the databases in which we will add these kind of facts, because these are not just basic, not some very basic trivial facts, but they usually follow quite complicated schemata. And we need the biocuration to design structured knowledge bases. So these include ontologies, which is a lot about my field. So I work a lot with ontologies, but other knowledge bases as well. So these can be RDF schemas and so on. And to add the definitions, labels, synonyms to ontologies or to classes in these knowledge bases. And in the end, also support users of these knowledge bases, for example, for information retrieval and people who want to use this information for machine learning or data mining. But now we have large language models, so a lot of things have changed, because when we look at what large language models already can do, maybe not as well in all cases as a trained biocurator, but catching up very, very quickly in many of these fields. So we can get text understanding and summarization. In particular, these large language models give us access to vast amounts of text So huge text corpora at the same time, and not just for retrieval, so we don't have to read this ourselves, but they can summarize a lot of text in a relatively small form, so we can consolidate even this knowledge. So this kind of compression, summarize some 20 papers or 5 books in a paragraph, and leads also to a consolidation, so the majority votes or some crystallization. And this is not very well understood how knowledge is distilled in a large language model, but it is a phenomenon that I think is one of the most powerful applications of large language models. And of course, we have seen this already, so they can extract entities from text or relations from text at various degrees, but all of these tasks that biocurators would be doing, large language models at least can do to some degree. And they can also generate text in particular, and you can try this. They can generate labels or synonyms based on some description for ontologies. This is very useful. They can also get extremely nice definitions for a class or, for example, for a disease. And they have some common sense knowledge. So this is so they can disambiguate by context, and they can perform some statistical inference, which means they can perform some inference even if some explicit facts are missing. So just statistically, they can fill in some gaps based on common sense background knowledge. And I have more, so they can do entity linking and resolution. There's a lot of work on entity linking. So they can essentially identify whether 2 names or identifiers in 2 different places refer to the same entity by context and so on. So this is in text. So this is named entity recognition and then normalization to some kind of identifier. But you can also think of ontology alignment or schema matching if you are more into databases. This is something that large language models can do, and they can do it sometimes better than humans in some domains. They can do event extraction. Event extraction is maybe a little bit related to relation extraction, but more complex kinds of entities. And they can also construct knowledge bases automatically, including ontologies. So maybe, again, not as well as a human, but this is something that can be argued for or against. So maybe Fabian had a strong opinion that this is not possible, but there is a lot of progress on this, and especially humans make a lot of mistakes when they create formal axioms. Or they can make a lot of mistakes when they create formal axioms, something that large language models can avoid. So now the question is, the question that I'd like that I ask is, of course, large language models have some problem. I'm not going to talk about these hallucinations, which make them limited in some safety critical applications. So they can make up stuff. And if you want to base a drug treatment or some treatment or anything, any decision that is actually relevant on this, okay, you don't want this. And explainability and interpretability. So this has been said many times. I want to focus on 2 other things. That large language models cannot yet do very well. So the first one is they don't have a way to explicitly represent knowledge. They don't build world models and reason over these world models. And this is something that curators can do. They can do very basic sanity checks. Does something make sense? So if you find the statement, there's an individual, female individual, and has a damaging variant in SRY, which is encoded on chromosome Y. So this is a protein, SRY, or a gene, and it's on chromosome Y. Then obviously a biocurator would say, oh, this does not seem to make sense. Large language models take this as given, and they just skip over this. And they can, if there are world models, we can test consistencies with existing knowledge. So this is something that is very difficult, in particular, to reason over knowledge that has been extracted with respect to what it already knows. So this kind of relation is something that's missing. And then there is maybe the most important part that large language models don't yet do, and this is they don't deal with long tail knowledge, which means you can actually check the performance, and this is a problem with many benchmarks, question answering benchmarks and identity recognition benchmarks. So when you check their performance, this works really well if it's a question which has many associated documents with the answer terms in it. But once you go to something that has very few documents, then their performance suddenly suffers drastically. So this will be they will be very, very bad on facts which are quite rare. So if you talk about a heavily studied, or you want to know something about a heavily studied disease gene, or anything, BRCA1, BRCA2, it will give you perfect information. But if you want to have some obscure gene in Drosophila that 5 people in the world ever looked at, then this is something that a large language model will not give you any reliable information about, while a biocurator will exactly give you this information, and this information can be used. So my question was, how can we find a way to maybe overcome these limitations? So how can we use the logical content of an ontology or a knowledge base together with text in a large language model? And how can we deal with this long-tail knowledge? And also dealing with study bias. Study bias here is that they are very good at things that are studied very well. So the way in which we approach this is using this neurosymbolic cycle. So we take knowledge, explicitly represented knowledge on one side, and combine it with data. I take text here also as data. In my group, we study various kind of relations, so genotype-phenotype relations mainly, but also protein sequence and protein function. And we have studied this kind of loop for a very long time. And I will give you an example of how this is going to work and what we can do with it, so that we can build models, including large language models, and endow them with an ability to reason over some of the knowledge that they have seen. So just for this, so we have knowledge on the left-hand side here. This is something that we can reason over using automated reasoning or some algorithms, many algorithms that we have built in computer science and related domains to reason over symbolic knowledge. And on the right-hand side, this is where we do pattern recognition, and large language models are really great on the right-hand side. And then they give us something that looks like knowledge, or they can give us something that looks like knowledge, but they don't lack this ability to reason over it and test whether it's consistent with other knowledge that they might have, because this knowledge is contained on the right-hand side as weights in some very large neural network. So most of what we have done in the past was to study embeddings and different types of embeddings of knowledge in some kind of data space. And I will show you what I mean by an embedding, and then I'll show you how to do the extraction so that we can do reasoning at the same time with some more modern embeddings. So an embedding is essentially just a morphism. So it's a map, a structure preserving map. We take some mathematical structure that we use to represent some knowledge, and we map it in some other structure where we can maybe do some machine learning better. So the whole idea is that the second structure is more suitable for some operations that we would like to perform than the first structure. For example, in particular, for applying some gradient-based optimization. And in the past, we have studied knowledge-enhanced learning. How can we endow a neural network with some background knowledge, some ability to, some knowledge-based capabilities? And there we wanted to take some knowledge base, some ontology, for example, and embed it in the RN so that we can apply compute gradients, and we can have a neural network use or access this knowledge in particular ways, so that we can preserve also not only access this knowledge, but also reason over this knowledge. And we have different approaches for this. And my group, I'm not going to go through this, but we have been doing this for a very long time, since 2017. We have created different embedding approaches in the past. And something that we are still doing now, and the latest version, and I'm just going to talk, I'm showing you a bit the latest versions in the last 3 minutes that I have. The latest versions have some very unique property, because I'll show you the first part. So the first part, so if we do a graph embedding, so we start with some ontology, then we project this ontology into a graph. So there may be different ways in which we can represent an ontology as a graph. Maybe we just take the syntax tree, or we have some other way, and then we use some graph embedding in the RN, perform link prediction, and then we get axioms back from this graph. So then we have inferred some axioms, we can apply reasoning. For the right-hand side, we have some methods. So graph embedding, knowledge graph embeddings method. For the left-hand side, we have a number of projection methods. But the big question is, what axioms should we actually infer by this method? So what are these axioms? And the way in which we see this, and I see already Susan, so I know that I'm almost out of time, but I understand I will not go very long because I understand I don't want to take anybody's time. But what we have done is we have taken one of the best studied relations in computer science, and this is called semantic entailment. And we have weakened this relation into approximate semantic entailment. I list here 3 different ways in which we can approximate semantic entailment. And here by approximate, I mean that under certain conditions, an algorithm will actually perform semantic entailment. We just weaken this so it may not never reach this particular state. We don't have enough time to do it. So with this, what we can do is, okay, I have EL embeddings, I have way too much time, I have way too many slides, but then we can do, for example, we take this embedding space, this generates a model, we can determine truth. We have many of these models, not all of them, because there are infinitely many, but we have maybe 10 such models. We test for truth in some kind of Rn. This is a space in which a neural network would operate and which optimization happens. Then we test for truth, and then we test for truth in multiple models in order to determine whether statements are true. So essentially what we have done, and this is my last thing, my last slide, what we have done with this is we have taken this knowledge enhanced learning and classification of knowledge, or with background knowledge, and formulated it as a semantic entailment problem that neural networks perform. So, and this is the last thing, and this is the last slide. I'm not saying that we don't need structured knowledge anymore. Structured knowledge will be is very important for some things, but there's one thing that has happened now, and this is that was a paradigm shift, and almost anything that Biocuration has done, we should revisit. Any workflow that we have, Biocuration workflow, that we already have, established ones, we should revisit in the light of large language models to see which parts a large language model can do better or can support a biocurator to do. And this is the big remaining question, what the role of structured knowledge in biology will be in the future. I'm very sorry for going over time. Thank you very much.
Thank you. That's okay. I think you kind of blew everybody's minds a little bit. I'm not getting a lot of questions. I do think that that last point of reevaluating workflows in the light of large language models is a key one that's on all of our minds. How do we make our workflows more efficient? How do we actually get these tools incorporated so that curator time can be spent to try to dig into the gigantic backlogs of data that we all are sort of drowning under at this point. I think at this point, if there aren't any questions from anybody in the audience, we might just take a 5-minute bio break, and then we'll come back for the panel, and I think we'll have a very lively discussion. I want to say thank you to all the speakers again. I really appreciated and enjoyed all of your talks this morning, and I'm looking forward to a great panel session. Thanks, and we'll see everybody back here in about 5 minutes.
So there we go. So we wanted to start with, for the 3 speakers, do you have other questions that came up during your talks or the others' talks you want to start now and discuss before we start the panel questions? The token question was a really good one. Are there other topics that you think are pivotal to kind of start us off with? And if you don't, that's fine. We do have topics here. So I know we showed you these a little while ago, but are there specific topics in this panel discussion set of questions you'd like to tackle first? Maybe something about the interactions between biocuration projects and ML development, how you'd like to see that moving forward? Are there areas where the Biocuration community could certainly be contributing more, or providing like beta testing feedback maybe, or attending your conferences? What do you think about how increasing this communication? Anyone on the speakers are welcome to respond.
Well, I can start off with, go ahead, Robert.
No, please go ahead.
All right, I can start off just briefly. I mean, with our tools, it is important to note that a lot of times people will find that it's, you know, some percentage of the way there, but then, you know, something else is needed. Yeah. And sometimes that's where the conversation dies. And so where we've been successful has been with sort of building a partnership where, okay, recognizing it's going to take us more than 2 weeks, sorry, to add the piece that you need, but also, you know, in the end, you should have something that would be useful. And so that's an approach that really quite helpful. Just as something to get started on talking.
Robert, did you?
Yeah, so maybe I have one point, because I have in my group also a biocurator, so I have daily, almost daily interactions with biocuration, and we have this discussion. So we have these discussions, especially now. So some parts of my group work on large language models, on different kind of predictive tasks, and then there's a biocurator, and of course there are students and so on. They like to sell their research, and they go in group meetings or in some other meetings, and they talk about what they can do. And now we don't need any biocuration anymore. And of course, it's overstating this. So they do text mining, and now we extract all these relations. We don't need this. We have function annotation we can do. We don't need, so we can do all of this very greatly. And it's true, we can do this so much better than we did before. On the other, so I get this overstatement, and we get a lot of overpromises, because a lot, when we look at these 0.9 F measure, 90% accuracy, 95%, we have all these tables. Many of these tables are quite biased. So it's very difficult to reproduce some of this. It's very difficult to generalize it. But then I get on the biocuration side, I get something else, because the obvious defensive posture is, no, it's never going to be possible to replace a biocuration task because it's so much better. Which also is not right, because of course biocurators will also, they're humans, they will also make mistakes. So what I would really like to see at some point for a databases are accuracy of facts in a database have different importance. So there are databases where it may be okay to have a few facts that are wrong, or it may be, or they might have an evidence code that might be of a lower threshold. Go functions are one big example for this. What I would really like to see is at some point where we can meet in the middle, and biocuration or database developers, maintainers tell me, we are fine with having 2% wrong knowledge in our database. With that, we are fine. Then there is a clear goal, not 100%, because this is not achievable by a human and not by a machine, but something below 100%. And then there is a goal that is being set, and we can see how far we are really between human performance and machine performance on these tasks. So some middle ground, I think, would be good to find.
Do you think, to all the speakers, that there's a sweet spot in this curation pipeline, knowledge generation pipeline, where we could have the machine learning? We're all using machine learning tools various places to inform our curation, but is there a sweet spot where we could identify it being a critical element in our workflow? But then at the other end, what I worry about, or I think about, is we have all these fantastic biocuration generated databases. How do we distinguish to our users everything to this date, or that has been generated through human curation, and then wanting to add in some of the machine learning data? How do we distinguish that so that it's very clear to users that they go to some website, assuming all of it's manually curated, but when we start adding things in that are developed by an algorithm, how do we demonstrate that to people so they realize there could be a % of error that's higher than 2%? So they're sure of the data that they're looking at. How do we share that so it's appropriately integrated and represented? What do you think?
So I have a suggestion for this. I'm sorry for jumping here, because currently we use evidence codes. Evidence codes are very bad for my students because I can see that every time they try, we do a lot of function prediction, protein function prediction. They go through QuickGo and download all the annotations. So they download the Goa database and download all the annotations. They don't know what an evidence code is. So they ignore all of this. And then they start getting a lot of predicted functions. Some of them are really good. Some of them are bad. We need something that is better than an evidence code. We need a lot more curation of how a prediction has been made and what errors we would expect. So if I go through QuickGo, I get this IEA, inferred by electronic annotation, Go functions. But an annotation that has been made by Interpro, I generally trust, or by PFAM, or by Panther, and an annotation by, I don't know, by some obscure protein function prediction tool, I might not trust as much. And I would really like to know how many errors am I going to expect. So I would like to be able to go, we can collect all of this, for example, for Go functions, but I would like to put a threshold on something easily recognizable, like a false discovery rate. I accept 10% incorrect Go annotations for whatever I want to do. And I should be able to retrieve exactly those where more or less we have a 10% error rate. So something like this would be really, really nice. And we can add this to databases, but we need a different standard. We need a new standard, a new way of capturing this type of information.
Yeah, I really resonates with me, the standard for the quality of the predictions that are being made. I also work in the genomics sequencing community, and we have standards for all types of genomic sequencing where we actually have these thresholds defined by the community, and that threshold of as a quality metric gets annotated with the primary data as it's put in. So yeah, it would be fantastic to be able to, integrate an algorithm, say this data came from it, and this is the threshold, whether it was reported on its initial publication or other publication. Yeah, I think that would be fantastic, because then it's very seen by the community, and knowing which data came in which format. And what, yeah, I think that's a great suggestion. What other speakers think of this? And other people that are on the session.
I'd like to second sort of that piece of the discussion, because it takes time with automated systems to become confident that, okay, that it's working reasonably. So you need to capture this, this prediction was provided by such and such a system. This subvariant, this particular model of the system. If it's ChatGPT, capture the date because it changes, right? If you want these things to be repeatable, it's going to need to include that information. And then the next question is, well, wait a second. Sometimes you have multiple systems that are very different. And if they start agreeing, then they're probably right. And so maybe you actually are looking for, that in and of itself becomes a useful signal that you can use, which I have.
But this is complicated when you have a dependency, right? So for example, for Cabby, I didn't mention it in the presentation, but the state of the art is classifier, and we try to compare our performance against them. But then classifier is used by the Cabby developers, and it's not clear from Cabby where this is used. So we couldn't really filter out during our evaluation which of the the manual signal distinguished from the classifier signal. So you really want to, I mean, if you want to have this kind of independent validation, you need to, from different systems, you really need to make sure that you track which systems have been, involved in the creation of the data for these systems. So it becomes more complicated. Another thing that I want to point out, I mean, it seems very important also to have not just tracking of the information when it was produced and with which version of which system, but also have the opportunity to retract information with information from the new system. I mean, for us, like Cabby, for our tool, the Cabify tool, we updated regularly with new models, which all, they are better. Otherwise, we wouldn't improve it or replace it. So if somebody would rely on prediction from us, it would be probably good to be able to remove previous predictions and update them with new versions, which could be more fine-grained, for example.
Solid questions. Sue, did you say you had a question?
Not so much on this, but related to the idea of how do we go, from these ideas, from the prototype tools. Like, I was struck in Robert H's talk about how many different models they've tried. It's like a model a year or more. They keep evolving, they keep changing. And I know from my recollection of the many times that MGI has worked with various groups to try to develop tools to support workflows, it always seems to fall apart at the step where we go from we have something that looks like it will work to trying to get it to integrated to a practical tool that's in our workflow, because that's what we need. And yet the funding for machine learning and the development of these tools is on the development side. That last integration step, we can't seem to bridge that divide very easily. And I don't know if the panelists have thoughts as to how we might advocate to funding agencies and to the different communities as to how we try to get over that what feels like a giant chasm.
What do people think? Suggestions, thoughts?
It's a concern, but but my initial response would be to ask, I'm certain there's a problem. I'm certain of it, but it's so many pronged that I'm not entirely sure what the specific issues that MGI has experienced are. And because I think that the problems might be just a little bit different, depending on whether you're talking about, okay, the accuracy is not good enough on this subset of the data, or if okay, it handles these tasks properly, but it doesn't handle this other one, or it's just not fast enough. We can't run it over all of our text, and we have to be able to run it at the point, and we just can't get the results fast enough for it to be usable. Those would have very different answers from my perspective. Some of those go ahead.
I was gonna say, some of it often comes down to, I think, trying to get just from the system and environment it's been developed in into our system and environment. There's always that challenge of the need to get to all the text and not just a part of the text. And in our case, it's not just all the text, it's all of the text in the main article and all of the supplemental materials, which are in, all these different formats. So some of it goes back to not necessarily our problem or your problem, but a publisher problem, which is a challenge for any of us to try to deal with.
Well, I think it's a generalizability problem too. I think you have tools you develop in my own projects that you develop for your own reason, and you go, oh, I want to make it general for the public to use, but you always have dependencies on things that you've encoded, right? You can't
Yeah.
write a code without dependencies in your own system. So how do we make them maybe a little bit more generalizable? Is that feasible to ask funders to allow that, or is that a path we could take?
Well, and we see that for the, as we move from the MGI system into the Alliance system, what works for the literature for one model organism does not necessarily work for the literature for another model organism. Just the way the papers are written are sufficiently different that the various tools start to fail. And yeah, how do we get a broader tool? How do we sort of find ways to make these things more integrated? Or if it's a specialist tool, bring it into the specialist system. Yeah, I have lots of questions. I don't have lots of answers.
On the development side, are there protocols, processes to define what these dependencies are? Is that like a standard aspect of developing them? Like to when you're kind of putting it out and publishing them or thinking about them, to define what those dependencies are. So if MGI wanted to take on a tool developed by Palmbase, see Palmbase on the Zoom, are we writing our tools, or could we ask each other to write our tools to define the dependencies, at least to then understand where things might be able to be connected or not? What do you think from the developer side?
Well, so there's two different kinds of dependencies that probably need to be mentioned. One is sort of software, hardware environment, and then the other one would be what other inputs does the system require, right? And so software, hardware dependencies, well, I mean, we've been working on that for many years. But yeah, there's a lot to be said for some of the recent environments and containerization and so on, where you can get a Jupyter notebook up and running for something you've never had running before very quickly. So that would need to be something that the developers would need to start paying attention to, and it certainly, having the right tools certainly helps. I mean, my own stuff previously was in Java. Now it's in Python. Much easier to be able to hand it over to somebody. The second half, yes, it would absolutely require some sort of specification, and I know that there are standards for this, specifying pipelines and that sort of requirements, but none are coming to mind at the moment. Apologies.
I wonder if there's sorry to dominate at all. I don't mean to, but I wonder if there is the opportunity to push it to funders, to ask them, say, when we are getting funds for developing tools, to have a component, a small component for getting things into a notebook, for generalizing them. Is there, because I know when we get funding to build tools, on my own fund, you get tools funding for your own system, but is there request we could put out to funding agencies, or a position piece to the community could say, says we also need this extra piece where we can actually have additional outside collaborators that we could then co-develop the product with after the fact? Is that something that we could all wish for?
Sure, but one thing that might actually be a little lower hanging fruit than that is publishers and reviewers. Make sure everybody knows, look, if you get a software paper to test, make sure that you can download it and run it yourself in a reasonable amount of time, and have that be part of the review. Is that that takes time, obviously, and it's not it, but yes, that the more we push on developers such as myself to make tools available in a really convenient format, the faster they'll be, the easier they'll be able to be adapted and integrated.
Yeah. So the community could come up with a standard and actually push that out to the journals and ask them to start promoting that. Even like you mentioned earlier, having the metrics of the algorithms, the models represented was part of that reporting, so that people know very clearly how the data was presented, what they're looking at for that system. that's something publishers do. We've done it throughout the genomics community, certainly. There's lots of examples of that, and ontologies and standards. So yeah, I think maybe the machine learning community needs a standards effort to be pushed out to that community, to the journal community, and ask them to start representing things the way you want to see them represented.
So there is one thing I wonder. Do you think, Lynn, that it may become one of the roles of a biocurator to identify which machine learning algorithm or tool would add value to the users of the database, the knowledge base that is being curated? Because there are some standard benchmarks, and there's a big problem with standard benchmarks because many times things do not generalize. I mean, standard benchmarks, especially very popular standard benchmarks, you don't even know if a large language model has already been trained on them. Probably yes. But do you think it would become one of the new tasks, or it could become one of the new tasks of a biocurator to test and evaluate different machine learning tools for generating content, to determine which of them are going to generate value for users of this database, and then add this information to the database?
Yeah, I think that would be fantastic. I know, I'm sure many people on this call have been doing exactly that as one-offs, right? We work with different collaborators, but wouldn't it be nice if we could have that more clearly shown in the community? Like on our own websites, is there a forum we need? Is there a place where we could attest or share the testing we've done as part of it to share it? Yeah, I think that would be useful. People could see what has been done.
And then there are some problems, because there are some common issues that occur. So, for example, in genomics, you will find a lot of duplicated data just by orthology, for example. So there are a number of things to consider when evaluating whether a machine learning tool actually works. Sometimes they work really well, but then you find out, oh, they're basically identical genes or proteins in the training and testing set, so no wonder they perform so well. Is there anything you think that the machine learning community can do, or people who develop these tools can do, in order to support also biocuration? Because this is something that is not trivial, and machine learning people are not very good at this, in my experience. I mean, they like to have very high accuracies and F measures and AOCs, not very low ones if they split their data differently. If it lowers that, that is not a good, that doesn't get you a paper at NeurIPS. So that is not a good, that is not a good thing to do. So it's also something that machine learners need to learn from people who actually care about that this data matters to their users. And this is not machine learners or machine learning people.
Yeah, I think a couple points. I'm going to just jump in here with that. One is, I would hope, and it's part of the reason we've been trying to encourage people to join the ISB Slack, is that we could use that workspace and that vehicle for communicating these issues, because for sure the communication back and forth and having channels that are open would really be very helpful. So I think I'd just like to put that plug in there, and now I've forgotten the other points I was going to say.
I think incorporating this kind of beta testing, or identifying, 5 people or less groups that you could actually, as a additional component to building your tool and publishing on it, but having a form of a way of getting community feedback that's positive and helpful for your development.
I think the other thing I was going to say is this seems like an area of unmet need for training, because you say machine learners aren't very good at that. I don't necessarily know that biocurators are great at knowing how to test a machine learning algorithm. They may have ideas about what matters to them for the data, but knowing about how to do that rigorously and in a way that's actually going to feed back well to you to make the tool better can be a challenge. And so there are a few different groups out there who are working on training and what training materials are needed. And maybe this is an area where we can say, oh, like I know Elixir in Europe is developing a biocuration group, they have a biocuration focus group. So maybe that's something we can say, well, this is an area of training that it would be really great if you guys could work up some documentation on how to do this. How do we actually make that happen and in a way that works for everybody?
Yeah, I
I
think
have a
it's really good. Go ahead.
So a couple of thoughts. One thought is Robert Horndorf is certainly right about what is it, these machine learning models tend to work really well on data that's similar to what they're trained on. And for a lot of the data sets, the testing set was randomly chosen. What is it? The split between test and train was just randomly chosen. And because of that, when you try to apply it to all of, all the biomedical literature, there's going to be subpieces that look completely different from what it's seen in the training data. And that's one of the reasons why the generalization problem there. Of course, there are ways of handling that, right? Realistically, we need to continue working on annotated data sets. By the way, if you have a ontology that you want that's NLP people to use, the fastest way to do that is to annotate a data set with it, and they'll start using it. Of course, that's also harder than it sounds. And there's things that you can do. For example, there are websites out there that allow you to take a data set and upload it as a test set, but you never actually allow the systems to see the final answers, even though you can give them their scores. And so that kind of protects your data a little bit. And to the degree that this is an issue, well, because it is always to a degree, the one looking at selecting data that will match your use case, either for a small quality assurance test set or a larger set that is released for the public, something so that you have confidence that it really is going to work reasonably well. And that it's almost that selection process that is critical. You don't necessarily even want to do it randomly. You want something that's representative of the sorts of articles that this system is going to be applied to. And unfortunately, the corpora that are out there right now, you read how they were selected. Certainly the older ones, you read how they were selected. It's not necessarily the best way to do it. Some of the newer ones we've intentionally tried to improve the selection process, but it's a tough question, trying to make sure that it's going to generalize now and 15 years from now.
Would it be of use if the biocuration community, through the ISB, was a venue for requests to be received? Saying, I'm doing this kind of modeling. Are there researchers out there that have data sets that I could collaborate with? Would that help machine learning development? Is that a reasonable step?
I think so, because there's a couple different pieces to that, right? Is there something out there already? Is there some machine learning person who's already interested in some specific task? Yeah, so there are different pieces of that question, and but yes, in essence, I think one of the thing that is most important is meetings like this one, where we're two sides of the same coin sort of thing, discussing the problem and trying to find areas of agreement and ways forward.
Yeah. Fabian, did you have anything you wanted to? And then looks like Val maybe is taking off her.
Yeah, I was just going to say, I know that as curators, we do have to review, obviously, the output of machine learning and AI projects. I've actually reviewed probably 3 things in the past month. And I just want to say we do have to think of a way that not only that we get recognition for this, but I'm the only curator at Homebase right now. I've done a project to review definitions, which was fine. reviewed a paper that made predictions that actually the only way you can review them is to trawl through them and then give feedback. It would be really good if I did really identify a curation group that their output is going to benefit before they start, because quite often I can't even see the purpose of the data that's been generated. It doesn't really fit into the schema of fair data. It's like, where is it going to go? Who is going to use it? I'm an expert in the data that was produced, and I had a real job understanding some of it. So I knew that nobody down the line would be able to do anything with it. And I just think it's really important that the use case and the end product and what it's going to be used for, especially if it's predictions. I think for entity recognition, it's very different. That's going to be useful to curators to find papers to curate. But I think sometimes there's a little bit of a misunderstanding of the kind of information that we're trying to extract. So yeah, I think there's a lot of bridging gaps that needs to be done between the two communities for sure. But no, how those would be done, but that's one of the things that we will do. You're right. Sue mentioned that the biocuration focus group that was set with Elixir, that's somewhere that we were hoping to do this kind of work with tool developers. So if anybody wants to sign up to that group, and also we've got lots of test data sets that are curated in the way that we would curate them. Like POMBASE has got 100,000 phenotypes extracted from 3,000 or 4,000 papers, all standardized data. Like, I can't imagine that any machine learning algorithm could reproduce that, but I would be very happy if they could, because I've got 2,000 more papers to curate. There's a challenge for you. But the scope of the project does need to be very well outlined to align with the curation and the database requirements. I think that's the point I was trying to make. Otherwise, there's no point. And anybody doing curation, certainly, Go function prediction, definitely reach out to the Go consortium, because we'll be able to give you pointers about what is a useful thing to do. And we have this big problem between what's actually unknown and what's unannotated, and that's largely because the number of curators is really small worldwide, less than 200 probably on every single project virtually, at least for the public databases or the core biodata resources. It's a really small number of people, and the more of this reviewing we need to do, it's squashing away at our ability to actually extract information from the literature. So yeah,
it sounds like there's certainly topics here for further discussion at another workshop. I know we're getting close to the hour and we'll be wrapping up. I think that's certainly a thought Sue and I had earlier, is how do we continue this discussion? Because I don't want it to end. We certainly think there's a lot of things to be covered. So either on the machine learning development side, do you think there's opportunities where we should be having biocuration individuals representing or bringing this topic up? I certainly think on the ISB biocuration side, there's certainly many things to still discuss. And I'd love to have other people in the audience to bring up points they're thinking about the last hour before we run out of time.
Maybe I just a very quick comment also to what Val has said. She said that there are many challenges and data sets that are being curated. So machine learning really likes challenges, because if we have some benchmark sets, it's very nice because we can compete. And you can look at challenges like CAFA. CAFA is great, and I don't know how many people competed in this year's CAFA. It was on Kaggle. So for computational assessment of function annotation for Go function prediction, but I think hundreds of different groups competed. So one way to engage with machine learning very closely is to bring a different set of diverse data sets to a machine learning meeting or to an AI meeting and pose it as a challenge. It's like, this is what we have done. And now, exactly as Val has said, now, can you reproduce it, please? And if somebody can, then okay. I would also doubt it, but That would be a very interesting challenge. It's like, this is what we have done, now reproduce this. So then that is one way in which you could engage with machine learning people, and they will be happy to have a new data set.
I can't stress what or underline what Robert just said enough. It's true, absolutely. I mean, mea culpa, computer scientists. I mean, if you ask what is the ontology of a computer scientist, it's strings and integers and floats. That's the world of computer scientists, and they don't care about data. I mean, in general, overgeneralizing, but it's true of many of those. And that's just unfortunately the case, and you have to live with it. But if you challenge them, if you give them a data set and say, here, you don't need to really understand what it does, and you don't need to understand what these strings mean, but try to outperform. That's where then that works very well.
Do you think
And it is hard to find good data sets. It's really, I mean, for the people who want to use. So, for example, we have this, pre-trained chemistry thing, and we wanted to predict something about chemicals, and we found this TOX21 data set, which has been used in dozens of papers. It's not a very good data set. It's a lousy data set. It's not large. It has many gaps, and the quality isn't very good. But it's there, so people use it. So the competition for you, as if you post a competition, if you have a reasonable data set, I think people will be very happy to use them.
One example of a location where that has been done is in the Biocreative workshops. Happen every other year or so. Currently, we've got Biocreative 8 underway. I just added a link in the chat, but yes, anyone who is interested in sort of running a challenge, that would be one place with already a healthy group of people who participate worldwide, and well, Biocreative 8's running now. Maybe you can join for Biocreative 9.
Yeah, I think that's an excellent suggestion. And maybe even at the ISB, we could have a challenge of gathering those data sets, so we could put one of these together. Yeah, Birgit, you're up. Your hand's up.
Hi, yeah. Sort of just thinking about at the end of this, many of you know me. I've been at EBI for 9 years, so in the public field. But the last 2 years, I've been in industry, and we face exactly the same issues. But there's an issue of confidentiality of the data in terms of working with external groups and testing new algorithms that may potentially be of interest. So I don't have a solution. I'm just throwing it in here. Maybe that's another topic we can explore. We now have quite a few, ex-academia, now in industry, members of the Biocuration community. I know when was the last year at the UK Biocuration conference, you had a panel discussion, and people like Jane and Pablo were on there, I think. That might be an interesting topic to explore, because we use the public ontologies, we use the public data. We just can't share the proprietary data back.
Yeah, I do think it's going to take many different types of conferences to get the communities out there and thinking about the different things that we could discuss. Maybe some white papers would help from our community that's generated from some of this, challenging people out there to get people thinking in this direction.
How do you decide where to host Biocuration, the Biocuration meeting?
It moves continent to continent each year. So last year was Europe. This year it's going to be in India. Next year it's in the US, and then it'll go back to Europe. And we put out a call for volunteers to host.
So co-locating it with a machine learning conference, having it as a workshop. So I'm thinking even conferences like NeurIPS, it's not related to NeurIPS at all, but they have a lot of bio-related workshops associated with it. And what would be really nice is to have simply is like, look, we're here, we have all these data sets, all of them are there, and give them to people and explain what they mean and what they are, and find a way to engage with them. I mean, it doesn't have to be NeurIPS. There are other maybe easier conferences that are accessible in an easier way. Biocreative is another one, but Biocreative already is a established community, and it's focused on text mining, as far as I understand. And Robert, please, I'm not trying to say anything wrong here, but if you want the broader community on machine learning, NeurIPS or ICML, all of them have structural biology, computational biology workshops. So all of them would be good targets to have a biocuration meeting for a day or two.
Yeah, I definitely think that. I also think, you know, the biocuration meetings, we always request people to submit ideas for workshops. And so that could easily be a way to reach out. I think a good question for that would be, how do you like create, what do you need to make a good test data set? Like what needs to be in there, what shouldn't be in there? You know, I know one of the things that we had learned along the way was, you know, for our literature, we often have the true positives. We often don't have the true negatives. So over the years, we've gone out of our way to get the negatives into MGI, because you know, in the past, we just threw those out. And Lynn is pointing out that we have 2 minutes left in the hour. So I will just give our panelists a huge thank you. And if there's any last comments they want to add, and I will put one last plug in for going and joining the ISB Slack so that we can hopefully keep these talks going. And I put a quick poll out there. I've never tried to put a poll together before, just to see if there was actually interest in having another one of these. Yeah, this is our second. We could envision in, you know, 6 months or somewhere in that range, trying to put together another virtual workshop. But I think both Lynn and I would appreciate that if there is a lot of interest in that suggestions from the audience for speakers and topics would be greatly appreciated, because we're trying to figure out who's out there and what's out there. And it would be helpful if other people could point us in the right directions. So any last words from our speakers?
Just thank you for doing this. This is really important work.
And we thank you
Yes,
for
and thank
coming.
you.
Yeah, and all of your time, and everyone who came online. I just want to mention one last thing. We had our last workshop. We had floated out the idea of putting out a white paper to actually try and call out all the issues on both sides of this. We will invite the speakers to join us on that. Soon I'll be putting that together, I think a first outline in the fall timeframe, and we'll get back to you with that. I think it's important that we raise these topics to our communities and get the message out there of what we think we need and where we think it can go forward. And kind of a call to arms idea. So you'll be hearing from us. And thank you again. Great conversations.
Thank you
Thank you,
very
everyone.
much.
Thank you. Bye.
Really appreciated it. Bye.
Bye.